Good afternoon, everyone. Thank you for joining us for the August 2025 edition of Lunch a la Carte, where each month we get a chance to hear one on one from one of the previous CART researchers, and we are thrilled to be able to continue that service in with the new Rotary year. We already have one scheduled for September, so we hope you will join us on September the 17th to hear from Dr. Wong, and we have others that will be coming on in the near future as well. Also, don't forget our monthly meeting of CART Conversations, where we have an opportunity for all of our volunteers to collaborate and hear new ideas and work together. And that is held once a month as well. And that will be tomorrow night, August the 14th. For those of you who are with us live today, I hope that you can join us tomorrow night at 5:30. And then again in September on September the 18th. And all of those dates are on our website. If you have not taken a look at our website lately, I encourage you to do that. It is a brand new website that just rolled out about 2 weeks ago. If you see something that is not working, a link or something that is missing, please let me know. We're trying to double check everything at this point. So I'm going to turn it over to our past president of the CART Fund Board of Directors, Bill Parker, who is a member of the Rotary Club of Alzheimer's Research, and he's going to introduce today's speaker.
And Dr. Allen, I wanted to let you know that when you received your grant, my wife and I were participating in the CART presentations to a number of Rotary clubs, and I still could not explain what it was you did. So hopefully today you're going to enlighten me so that I fully understand what it is that we granted money for you. So with that, Dr. Allen is a British neuroscientist. She studies the role of astrocytes in brain development. Bear with me a moment here. Homeostasis and aging. Her work uncovered the critical role these cells play in a brain plasticity and disease. She is currently professor at the Salk Institute for Biological Studies and the Hearst Foundation Development Chair. She started her own lab at the Salk Institute and is now working to understand how astrocytes regulate synapses throughout life, both in health and in disease. She conducted her undergraduate studies in anatomical sciences at the University of Manchester in England. She completed her doctoral degree in neuroscience at University College in London, in the United Kingdom, in the lab of David Atwell. She was a postdoctoral researcher in the lab of Ben Barnes at Stanford University in California. Her research focuses on how astrocytes regulate the synapses in the brain during disorders such as Alzheimer's disease. In 2012, while she was a postdoc in the lab of Ben Barnes, she showed that astrocytes secrete glypican 4 and 6, which is needed to create the glutamic synapses between neurons. She later expanded the research on glypican 4, showing that it is needed for the postsynaptic neurons to receive inputs. Allen also showed that astrocytes excrete a protein called CHRTL1, which helps the maturation of the brain. It also increased neuroplasticity in the brains of mice. She uses ribotagging, which is a molecular technique to determine which proteins are made by the ribosomes. This technique allows her to show that astrocytes make a protein that encourages the breakdown of connections between neurons. And in 2018, Dr. Allen received a CART grant for $112,500, and her focus for this grant was to specify exactly what the astrocyte does in the brain, both in a healthy brain and the brain facing Alzheimer's disease. And without further ado, I would like to welcome now full professor, Dr. Nicola Allen. Thank you, Doctor.
Thank you so much. That's a lovely introduction. Hopefully by the end of today, you'll understand the project. And it also means that when I came to visit you all to receive the award, perhaps I didn't do a good job of explaining. So let me share screen. Okay, are you seeing the presentation? Excellent. Okay, so as was mentioned, what my lab is very interested in is understanding how the brain forms and functions and changes in different neurological disorders. And before I get into the topic of our grant, thinking about these cell types here, the astrocyte, and how they could play a role in Alzheimer's disease, I just want to kind of take a step back and give you an idea of the scale that we're studying this at in the context of the brain. So we can think about the brain at many different levels. Hang on. Oops. Let me just move that out of the way so you can see the top there. Am I still sharing? Okay. Sorry. It's a while since I've given a Zoom presentation. So this is just thinking about us, humans. This is the brain. We can think about many neuroscientists study this at the level of the whole brain, but we kind of think about what if we take a step inside here and look at all the cell types that make up the brain, and these are the neurons. And within the human brain, there are billions of these cells called neurons, and these are the electrically excitable cells that control our thoughts and actions. And the way that they do this is by communicating at specialized junctions shown here called synapses. And there are trillions of these in the human brain. And if we take a closer look at what this synaptic connection is, as I said, this is a connection point where neuron 1 sends a signal to communicate to neuron 2, and each neuron has thousands of these. And it's the balance of how these synaptic connections are activated that kind of controls how we carry out movement, for example, or how we interact with the world around us. And in addition to being a place of communication, this is also a place of learning. So a change in the number of these connections that neurons have, or in the strength of these connections, is how we can learn things, or also when they get weakened, how we forget things. And this is important in the context of Alzheimer's disease, because we know from studies from many groups that one of the earliest changes that happens in Alzheimer's is that you get actually a decrease in the number of these synaptic connections that the neurons have. So before you see actually an overall change in the number of neurons in the brain, the neurons actually become less connected and less able to communicate. So this is what we're interested in understanding. Can we actually prevent this from happening, for example, in the early stages of Alzheimer's, so maintain the ability of the neurons to communicate, or at a stage when it's already occurred, can we actually repair this? So could we encourage the neurons to form synapses again and actually improve functionality, even in the context of Alzheimer's? So the way that we do this, as was mentioned in the introduction, is we think about all the other cell types that the neurons exist within our brain. And this is just a cartoon of the neuron, so the electrically excitable cell, shown here again with this synaptic connection. And actually, when we think about the human brain, while there are billions of neurons, there's also billions of other cells present too, and these are called glia. So neurons are actually only half of your brain, only half the cell types that are present. And there are specialized glial cells that have very important functions. For example, this cell called an oligodendrocyte, this cell makes a lipid-rich membrane called myelin that wraps around the neuronal process and allows for very fast neuronal communication. So this is very important for allowing the neurons to communicate rapidly. The brain also contains lots of blood vessels, and this is important because the brain uses lots of energy in order to carry out the neuronal function. So by having all these blood vessels here, this is a way that nutrients and oxygen can be delivered at high levels into the brain to give the energy for this to happen. The cell type that we're going to talk about today are called the astrocytes, shown here, and they're actually named because of their star-like morphology. So astro is star and cyte means cell. And in this cartoon, you can see you have the cell body here, and there are many, many processes that reach out into the brain. And by doing this, they're interacting both with the neuronal synapse, so the synaptic connection, and actually sensing how the neurons communicate. But they also have this important position where they touch the blood vessel, and by being on the blood vessel, they're the cell that takes up the nutrients, such as the sugars from the blood, processes them, and gives them to the neurons to provide energy. And then finally, another cell you may have heard of from other grantees are the microglia. This is another type of glial cell. So these are actually part of the body's immune system and very important for fighting off pathogens and kind of protecting the brain from infection. But one thing that they also do, which can become important in the context of Alzheimer's, is they can also eat parts of neurons, including these synaptic connections. And there's some evidence that these cells can be eating too many connections in the context of Alzheimer's. So this is something, again, that can be a target for trying to improve synaptic connectivity. So that was kind of a cartoon overview of these different cell types in the brain. So now we're going to focus in on the astrocytes. And here, I just wanted to show you what one of these cells really looks like. So in green, this is an astrocyte. It's been filled with a green dye. And you can see here again the cell body. And these bright processes here are the primary branches. And then kind of all these smaller branches here are like the leaflets going out into the brain. And you can't see it in this image, but actually what they're doing is interacting with many different synapses. And you can see from here in red, this is one neuron that's also been filled with a red dye. And you can see that this astrocyte is completely surrounding and wrapping this neuron cell body and also the processes and branches of this neuron. So we know that these cell types are in very close interaction in the brain and have an ability to be sensing and responding and talking to each other. So what do these cells do? Well, they have many, many important functions that we would consider ways of allowing neurons to work properly. So I already mentioned their ability to take up things from the blood to provide energy to the neurons to allow them to function properly. Also, when neurons communicate with each other, they do it by releasing neurotransmitters, and they release quite a lot of them. And if they're not removed from around the neuron, then the neuron can't have another communication. So astrocytes have properties that actually take up or recycle these neurotransmitters from outside the neuron and then allow the neurons to keep communicating. So in that way, they're kind of maintaining the neuronal environment to allow the neurons to function correctly. The things that we've been interested in the lab were the more newly discovered functions. I say newly, it's probably about 25 years ago now, but really showing that in addition to having these very important functions, once the circuit has formed, we actually know that when the neurons are forming connections with each other in the developing brain, astrocytes are involved in this by producing specific signals that increase the number of synapses that form and also allow the neurons to communicate correctly. So what's the evidence for this? So this comes from experiments done, as I say, about 25 years ago, where researchers took a simple system. They said, well, what if we take neurons or astrocytes out of the brain and look at how the neurons function? Is it different when they're on their own, as shown here, or when they're in the presence of these astrocyte cells, shown here? And what was remarkable is that they found that if you look at the number of synaptic connections that these neurons form, and that's shown in the cartoon by these black dots, or in these immunostaining images here by bright green and by bright red clusters. When the neurons grow with astrocytes, they make many of these synaptic connections, shown by these bright clusters here. If the neurons grow without the astrocyte cells, so the neurons are alive, they're healthy, they send out the process that you can see here, they don't really make any of these synaptic connections. So what this tells us is that neurons don't have all the information they need to make synaptic connections with each other, and that astrocytes are a cell type that tells the neuron to do this. Second, if we then ask, well, are these connections functional? So are the neurons able to communicate with each other? We do that using a technique called electrophysiology, and what this essentially allows us to do is to record the electrical activity of the neuron. And what we see is that the neurons here that have been growing with the astrocytes, you see these kind of downward deflections here, and this tells us this is a synapse being activated, so this neuron is communicating. However, if we look at neurons, again, that have been grown without any astrocyte cells, we basically see this flat line. So as I said, the neurons are healthy, they're sending out processes, but they're failing to make functional contact with their neighbors. So telling us that neurons on their own can't communicate with each other, and they need astrocytes to tell them how to connect and how to communicate. And the work of my lab and many others in the field has really been to understand, well, how do astrocytes do this? What is the way that they're doing it? And what we now know is that there isn't just one signal that astrocytes release. As was mentioned in the introduction, we've identified a number of different signals, mainly proteins, that the astrocytes make, including the glycogen 4 and 6 that was mentioned in the introduction, that are able to tell the neurons to make connections and make them communicate. And that's really kind of the basis of what I want to talk to you about today, is asking whether we can now use this knowledge of how astrocytes in a healthy brain can actually tell neurons to make synapses. Can we use this in the context of Alzheimer's? And also, if we look in Alzheimer's, do these signals change? So do we lose them? And could that be some of the reason why the neurons become less communicative? So before I get into the actual project, I just wanted to give you an idea of why some of the reasons why researchers began looking not just at neurons, but at other cell types in the brain in thinking about neurodegenerative disorders, including Alzheimer's disease. And what's here is kind of this diagram here is work from a lab at UCSF, where they looked essentially at all these different neurological disorders that happen across our lifetimes. And they asked, if we look at cell types in the brain that either change in these disorders. So in studies where people have been able to take samples from brains post-mortem and look at the cell types and how they change, can ask which cell types change the most, but also if we, for example, think about in Alzheimer's disease, we know, for example, we can have more risk with things like ApoE4, so a gene called ApoE4, and we know that this is something that in the healthy brain is mainly expressed by astrocytes, and then in Alzheimer's also by microglia. So essentially what they did is they combined which cell type changes in these different disorders and which cell types normally make things that we know give you an increased risk of having this disorder, and then color-coded them. The darker red you see here basically tells us across all these different disorders, microglia and astrocytes are more likely to be changing or more likely to be involved. And everywhere you see this star, this means it was significant using statistical testing. So this is just to say, to give you kind of a visual idea, if we're thinking about different neurological disorders, including Alzheimer's and other neurodegenerative disorders, we should be looking not just in the neurons, but also in these glial cells as well, to have ways of coming up with potentially new therapeutic targets. So the overview of the project that we proposed to CART a number of years ago, and I'll go over with you today, was really asking whether we can use this knowledge of astrocytes to prevent synaptic dysfunction in the context of Alzheimer's. And we've done this in a couple of different ways, and really thinking of this idea that we know if we go from a healthy condition where the neurons have many of these synaptic connections shown by these black dots and healthy astrocytes in green, as Alzheimer's disease develops, we know that the neurons lose some synaptic functionality, and also we know that the astrocytes are changing. But we didn't really know how they get there. We know from many studies that when we get to kind of progressed stages of Alzheimer's disease, astrocytes have changed a lot, but we wanted to know how do they get there? So what's the time course of how they could change or how they could be changing early as a way that we could intervene? And I'll show you today work we've been doing using mouse models, but we've also started doing work now that I can summarize at the end using human cells coming from patients, donors who have sporadic Alzheimer's disease. And the two things that we've been doing is we've been asking, okay, if we can identify how the astrocyte changes in the course of Alzheimer's disease, can we actually reverse this change, so stop it happening, so keep the astrocyte healthy and then keep the neuron healthy? And our second approach, which I'll summarize at the end, has been to ask, well, if we kind of ignore how the cells are actually changing, but just think about what we know about what these cells do in the young brain and the healthy brain, can we re-express some of these signals we know would normally make synapses in a healthy brain in the Alzheimer's context and see if that has any benefit? So that's the second part that I will talk about. So yeah, as I say here, the summary, the goals of this grant were to ask, do changes to astrocytes allow synapse loss in Alzheimer's? And this is what I'll cover first. And this has been a project led by a senior postdoc in my lab, Isabel Salas, shown here. So as I mentioned, if we're thinking about the progression of neuronal alterations in Alzheimer's, shown along the top here along a timeline, while much focus has been on the loss of synapses that happens, which eventually leads to neuronal death, what we also know from many recent studies and also from patients is that early on, you can actually see an increase in neuronal activity happening. And this can manifest as seizures. And this is something that can actually happen early before you lead to the loss of synapses and neuronal death. So we were very interested in asking, can we actually identify changes in astrocytes that are happening at these different stages where you have this early neuronal hyperactivity or dysfunction, then the loss of synapses, and then the neuronal death. And on the bottom here is also just a timeline of what people have shown or think is happening in astrocytes across this timeline. So if the astrocytes function also changing, they take on this state called astrogliosis or reactive astrocytes that you may have heard of. And then by the end stage, they start to become inflammatory and communicating with the microglia and perhaps actively damaging the neurons. So we wanted to know, using animal models, could we actually identify across these different stages how the astrocyte changes and the level of things that it's making. And this was helpfully already introduced in the introduction, this idea of this ribotagging approach. So basically what we wanted to know is, in the context of a part of the brain that's involved in Alzheimer's disease, in this case, this is the hippocampus shown here. So this is just an overview of the hippocampus at very low magnification. If we zoom in, we can see astrocyte cells shown here in green. Excuse me. So we wanted to know specifically, how do astrocytes change their function in the hippocampus in these Alzheimer's models? So to do this, we use this ribotag approach, which essentially allows you to make all of the ribosomes in an astrocyte have a tag on them. And this tag is green, and that's what's shown here. So what's important about this is the ribosome is the place in a cell where you're actually taking the information from the genome, so the RNA, and then making it into a protein, so giving it function. So if we can tell what's happening in these ribosomes using this sequencing approach, then we know what the cell is doing and what it's making, and gives us idea about its function at this time. So we use this approach where all of the astrocyte ribosomes have this tag shown here. So then we can just break up the whole tissue and use a technique where we have what's called an antibody that has an affinity for this ribosome. It pulls it out as a mixture to isolate it. So you just now get the mRNA it's making its message, and you can use a technique called RNA sequencing to say, what is the cell doing at this time? And when we do this, what we can then ask is, well, once we've taken our astrocyte sample and compare this to the total hippocampus RNA, do we actually see an enrichment for astrocyte genes, which is our goal? And here, these pink line here, this is just a list of genes that we know astrocytes make. And by seeing if it's kind of a redder or an oranger color, that means it's enriched in the sample we've isolated, compared to the total input, which has these blue colors. And you see all these orange and red colors for the astrocytes, saying astrocyte genes are indeed enriched. But if you look at the other cell types in the brain, such as neurons and microglia and oligodendrocytes, we see blue colors for their genes, telling us we've depleted these. So this is just our way of saying these samples that we're analyzing are indeed enriched for astrocyte genes. So we can now ask, how does an astrocyte change in the context of Alzheimer's pathology? And we did this by looking at two different mouse models that represent two different features that can be seen in the pathology of Alzheimer's disease. The first is an APP mouse. So this produces beta amyloid. So it produces beta amyloid plaques. And what we can see here is, if you look across different time points in the mouse model and stain for these amyloid plaques in red, you see by this 12-month time point, there's lots of red. So there's lots of amyloid plaques present. At 6 months, there are some plaques beginning to form, whereas at 4 months, our earliest time point, we don't see evidence of any of these kind of aggregates of the amyloid plaques. If we then look at astrocytes, this marker called GFAP. This is something that marks reactive astrocytes. What we can see is that early on, there isn't much change to the cells, but by this late stage, where you see many of these plaques present, you also see kind of this thickening of the process and the cells look larger, telling us that they're in a reactive state. The second model that we looked at is something that is representing tau pathology in the Alzheimer's models. So this is kind of inside the neuron where you get the aggregates of this tau protein. And again, we can look at the different time courses and see that at an early time point at 4 months, there's not much red stain here. So red is now staining for this tau protein. Whereas by 12 months, you see lots of these inclusions or aggregates inside the neurons, telling us that pathology has developed. And again, the astrocytes are also, at least by this GFAP marker, also changing along with this progression. So these are the time points that we looked at to say kind of early before pathology, moderate pathology, and strong pathology. And we did this in both male and female mice to see if there were going to be any sex differences that we could detect. I would say we saw some sex differences in the amyloid model. I don't have the data here today, but a lot of this was related to inflammation. So that's something in the future we're interested in following up on, because there can be increased prevalence in female compared to male for Alzheimer's. So whether astrocytes could be involved in some of those differences. So the first thing we simply asked was, if we look at our different time points for 6 and 12 months in the amyloid model or the tau model, how many changes in these genes do we see happening in the astrocytes? And what was quite remarkable in both the models is that early on, at least kind of in a population as a whole, we didn't see many changes happening that at a statistical level using the statistical criteria we used. But by the time we get to 12 months, where I showed you in the previous images, you can see the pathology. There are hundreds, up to 1,000 genes that are changed in the astrocytes in these different models, suggesting that really they are responding to these pathology changes that are happening, and perhaps in ways that could be negatively impacting the neurons. So we wanted to know, well, this is a lot of genes. If we then categorize these genes based into groups on what they're doing, can this give us some insight into how the astrocytes are changing at these late stages in these models? And we do this using something called reactome pathway analysis. And essentially what you can see here, so red is all the upregulated pathways and blue is all the downregulated pathways. And you can see many of these things that are upregulated in both models are related to the immune system and inflammation. So things like complement, interferon, lymphoid cells, chemokines, these are all things that would suggest that astrocytes have taken on an inflammatory state that could be damaging to the neurons. And we see this in both amyloid and tau. If we then look at the downregulated, many of these things that are down are actually things related to energy production or support of the neuron that the astrocyte would typically be doing in health. So these things like respiratory electron transport complex 1 mitochondria, this is all ways that the astrocyte would normally produce energy. And we see that this is being decreased in both amyloid and tau situation. Another thing I didn't mention earlier, that something very important astrocytes do, is they actually make a lot of lipids in the brain, including cholesterol. And while we may think of cholesterol and the rest of the body as perhaps being bad when it's at high level, in the brain, cholesterol is really, really important actually for neurons to function properly and allow the neurons to communicate with each other. So we think it's very interesting that actually the ability of the astrocyte to make this cholesterol is being downregulated in the context of Alzheimer's pathology. So the combination of these changes really suggests that there's a gain of function of things that could negatively impact the neuron, and also a loss of function of things that would normally be supporting the neuron. We were then interested in knowing, well, are any of these changes the same between the amyloid pathology and the tau pathology, or are they unique? So we can look at that by asking on the level of individual genes, how many of them overlap between the two different conditions. And you can see it's quite high. So most of the changes we see in the amyloid condition are also happening in the tau condition, shown by this overlap here. And this is true for both the up and the downregulated. And if we plot these two kind of conditions against each other, what we see is essentially most of the genes that change change along this same axis here, telling us if it goes up in the amyloid condition, it also goes up in the tau condition. And in this box here, if it goes down in the tau condition, it's also going down in the amyloid condition. And again, just looking at these common pathways, really highlighting this upregulation of immune signaling in these astrocytes in these conditions. So overall, this kind of gives us a picture of how the astrocytes are changing. And also this fits very well with recent studies that have come out from many groups where they've actually been analyzing this in human postmortem samples and seeing astrocytes taking on these pro-inflammatory states and also downregulating these neuronal support states. So we think what we're seeing in these animal models where we can now test things to see what they're doing is actually really the same as what's happening in the patients as well. So it kind of gives us some validity to the models that it's worth moving forward and testing what some of these changes are. So for the rest of the talk, I want to focus on these factors that I've mentioned before that astrocytes make, shown here in this list, 1 through 5, that actually promote synapse formation and promote synapse function. And this is just some of them listed here, different things an astrocyte makes, like cholesterol, these glycogen proteins that allow a neuron to function correctly. And in the next part of the study, we wanted to know, well, do any of these actually change in the astrocytes in the course of Alzheimer's? And what I'm showing you here, these are heat maps where we're looking in the 2 different models, the amyloid model and the tau model, at the 3 different ages again, so the early, mid, and late pathology. And what you see is everywhere that it's blue, this means that this gene has gone down in the Alzheimer's context. And everywhere that it's red, this means that this gene has gone up in the Alzheimer's context. And we group them into genes that normally promote synapse formation. And you see the vast majority of them become blue. They're downregulated in progression. And then those that promote synapse elimination, and you see the vast majority of those get red. So they go up with the Alzheimer's progression. Again, suggesting there's now been this switch in what the cell is doing, so it's no longer promoting formation of synapses, and it's now actively promoting their loss. So given our expertise in the lab of really studying the formation of these connections, we wanted to focus in on things that we know are normally promoting synapse formation, but that get downregulated progressively, both in the context of amyloid pathology and also tau pathology. And when we look for those things that are common, which are shown by stars, this means significant by statistical criteria, you can see that there's this one gene that pops out. This is something called GPC5, which encodes a protein called glypican 5. So we're very interested in this as a potential candidate. And this gets to the second part of the talk, asking whether these astrocyte signals can actually be used to repair damaged synapses in Alzheimer's disease. So what is glypican 5? So as was mentioned in the introduction, when I was a postdoc, we identified these molecules called glypican 4 and 6, which are used in the young brain in order to promote synapse formation. But we also knew from our work at this time that there's not just these 2 family members, glypicans 4 and 6, there's actually this large family of these proteins, actually 6 of them are present in us, in humans, and many of them are made by astrocytes in the brain. And we found that while these 2, glypican 4 and 6, are present very early, so when neurons are first forming synapses, glypican 5 is something that actually comes up later and is involved in maintaining stable synapses. And we were able to show that by making a model. So this is a mouse model that doesn't have any glypican 5 in astrocytes. So it has it in other cell types in the body, but not in astrocytes. And we can look at this and ask, when we get rid of this factor just in astrocytes, does it change how synapses form? So here, what you can see in these images, these are stainings, very zoomed in, looking in the cortex of these mice. And we stained for something that marks the presynaptic side, VGLUT1 in purple, and the postsynaptic side called GLUA2 kind of in this blue color. And what we've done is we've counted everywhere that these puncta colocalize, and this is a synapse, so a place where the neurons communicate. And what you can see is that when you have lack of this protein just in astrocytes, you have less of these synapses forming. We could then also look at the size of these terminals, so the size of the connections, which tells us the strength and the ability of these neurons to communicate. And we also see that the size of these connections decreases when we lose this factor just in the astrocytes, telling us essentially that in the healthy brain, this is something that's normally maintaining synapse number and maintaining synapse size and functionality. So suggesting when we lose it in Alzheimer's, maybe this is contributing to the loss of synapse number and function that is seen. And the other criteria for us to follow up on this was to say, okay, well, I just showed you this data in a mouse. What happens in human patients? Do we actually see this factor changing in Alzheimer's disease? And this is a table that summarizes many recent sequencing studies that have been done from patients either with Alzheimer's or other disorders such as frontotemporal dementia and also multiple sclerosis, where they analyzed astrocytes specifically and looked at the levels of genes that they make. And what is seen in all of these different studies is that there are specific astrocytes where glypican-5 is downregulated, telling us that this is something that's also happening in human patients. And then we wanted to know, well, can we actually see this? So everything I just showed you was looking kind of at sequencing. So what we can do is we can use a technique where we actually go in and look at the level of the RNA for glypican-5 within cells in the hippocampus of these mice. So here, what you're seeing is a very overview of the hippocampus, and white is this glypican-5 signal, and blue is the nucleus of every cell that's present. If we then zoom in and look at the glypican-5 and ask what cell is it in, so the purple, these are astrocytes, so you see the white signal is present essentially within all these purple cells, which are astrocytes. What we can also do is label the amyloid plaques. So the plaques here are shown in green. And what we see is that those astrocytes that are touching an amyloid plaque, so close to pathology, these have lower levels of glypican-5 than those astrocytes far away from the plaque, kind of in contact with more kind of the healthier neurons. And this is quantified here, telling us that astrocytes touching plaques have less of this beneficial signal than astrocytes far away from plaques. And I should say, this isn't just something that we see with glypican-5. Other groups have shown this happens with other signals as well that are normally beneficial for the astrocyte function onto the neuron. So there's something about the plaque that's making the astrocytes stop doing these good things. So finally, we wanted to know, well, can we do anything beneficial with this? If this is something that's normally going down in the course of the Alzheimer's pathology, if we overexpress it in the astrocytes in the brain before we see these synaptic changes normally occurring, can we stop it happening? So can we prevent these synaptic changes with an astrocyte rescue, essentially? So the way we do this is using a technique using a virus that allows us to express glypican-5 or a control protein just in astrocytes. And we can actually deliver this brain-wide. So in this image here, this is a section of the whole mouse brain. This is the hippocampus here, which we were talking about before. And everywhere you see purple, this is where an astrocyte has overexpressed this glypican-5 protein. So you can see with the delivery method we're using, this virus, we've been able to target astrocytes across the whole brain. And if we go in detail and look in the hippocampus, you can really see when we stain for this glypican-5, this amazing intricate morphology of the cell. So it's really filling the whole cell. And with this model, we can now do a number of things. We can ask whether this impacts synaptic function changes we know happens, if it impacts learning and memory that happens in this mouse model, and if it changes synapse number or also production of amyloid plaques themselves. So the first thing we looked at is, as I mentioned earlier, in these kind of very early stages before you see synapse loss, both in humans and also these mouse models, what you actually see is a hyperactivity of neurons. So here I'm showing you again, like I showed you earlier, these electrical recordings of the neurons, where every time you see one of these downward deflections, this is the synapse being activated. And the wild type here is in black, and this is the amyloid mouse in red here, showing that there are many more of these deflections happening. And this is shown in the graph here, that in this amyloid model, the neurons have more activity, and this is bad. What we can then do is ask, well, if we overexpress glypican-5, what happens? And actually what we find is that in this kind of pink color here, this is the same amyloid mouse that's been overexpressing glypican-5 for 2 months. And you now see there are less of these synaptic activations occurring compared to this example here. And when we quantify this across all of the recordings that we did, we see that we've now essentially normalized the synaptic activity in these neurons back to what we see in the control condition, telling us that by overexpressing this factor in the astrocytes that we know stabilizes synapses, we've actually been able to prevent this hyperactivity from happening. So we thought this was good, this was great. We'd now look at whether this, if we continue the expression out to 6 or 7 months, when you start to see behavior changes, would this have any impact? So we looked at this using an assay that essentially it's called the Barnes maze, and this is a way that you train a mouse to learn how to escape from a platform. So what happens is it's kind of this very big ring, this platform in a brightly lit room, and the mouse doesn't like bright light, so it wants to kind of get back into its escape hole and kind of get away from the bright light. And it learns where this location of this hole is, where it will get back into its home cage, by looking at spatial cues around the edges. So essentially the mouse is looking and it says, okay, I see the square here, circle here, triangle here. I know that where I want to go is close to this circle. So you train it over a number of days, and what we looked at is how long it takes the mice to learn where the location of this hole is. And the quicker they learn it, that tells us kind of the better spatial learning that they have. And essentially what we can do is we can look at over this time, so over this course of 4 days, and also this reversal, how long it takes the mice to find the location of this hole. And the black line, these are the control mice, and you see they kind of learn over time. By this trial 6 here, they pretty much learn the location. Whereas the solid red, this is the Alzheimer's mice, they take longer, whereas the dashed red, which is Alzheimer's mice of glypican-5, they have this rapid learning here. And we can look at this in a different way, where we just kind of ask how quickly do they learn between day 1 and day 2. And we can see that the Alzheimer's mice, they don't really do better than chance in learning where the location is to escape, whereas the wild type mice do. And the Alzheimer's mice with the glypican-5, they more rapidly learn how to get away from this condition, telling us that by overexpressing this factor in the astrocytes, have this very mild improvement in their behavior, and now they learn more rapidly. But I want to point out, everybody gets there in the end, so it's not like this is a complete change. The Alzheimer's mice also eventually learn the location, it just takes them longer. And the final thing we looked at was, well, I kind of showed you this behavior change. We wanted to know whether this has any link to these receptors that we talked about earlier, these GluA2 receptors, which are shown here in green, which are present in the post-synapse, so the receiving cell, the receiving neuron. And what we indeed see is that when we have mice that have more of this glypican-5 protein, which you can see here in this green immunostaining, there's essentially more staining for this receptor, which is quantified here, telling us that one way this could be working to improve the learning is by increasing the amount of this receptor that the neurons have. And of course, we wanted to know whether this has any impact on pathology itself, on the amyloid plaques, and we actually found it didn't. So the improvements that we see in the behavior and in the synapses are happening despite this increase in amyloid plaque load that's occurring. So we think this is kind of overruling essentially the amyloid pathology to allow the neurons to keep some functionality. So this is a summary I want to tell you about, which really came out of this CART project, of where we asked how do astrocytes change across pathology progression in these Alzheimer's models, and picked up on one factor, this glypican-5, as something that's downregulated with both amyloid and tau pathology. And we showed that when we overexpress this or stop it being downregulated, then we can actually have some benefit in terms of it maintaining synapse function and also improving some behavioral deficits. So we're very interested if this will be beneficial in other disorders, given it's been seen in human patients, it's also downregulated in multiple sclerosis and also frontotemporal dementia. So finally, I just want to show you how our lab has expanded in the context of Alzheimer's from when this started in 2018, because this was actually the first grant that we got for Alzheimer's, so really launched us into this area. And this is just some examples by Ashley and Axinia of using different proteins that we know astrocytes normally make that are beneficial to synapses that also go down in the course of Alzheimer's pathology, such as pleiotrophin and cordon-like 1. We have evidence also that overexpressing them in the same way I just showed you for glypican-5 also has some benefit for synapses and also learning and memory. We're also interested in the other side of the coin. There are some factors such as this protein called IGF-BP2 that gets upregulated in Alzheimer's, and we think this is a negative thing. So we're asking if we stop this upregulation, will this have some benefit? And as I mentioned, we're also very interested in having using human models in our experiments. So we've been looking, making astrocytes actually from induced pluripotent stem cells from patients who have sporadic Alzheimer's disease, and actually identifying in these patient-derived astrocytes that there are intrinsic changes that happen in these cells that could be negatively impacting the neurons. And we're very interested in what those changes are, how we can target them, and how we could have some benefit in these models that come up with new therapeutic approaches for Alzheimer's. So as I said, just sort of finish by acknowledging my lab here at Salk. I have an amazing group of current and former lab members. Obviously, I want to acknowledge you for our first ever grant on Alzheimer's that got us started in this area. That then led to more funding from Cure Alzheimer's, Chan Zuckerberg Initiative, American Heart Association, Nomis Foundation, to name a few. So it was really kind of, as was the goal, the starter grant that got us started in this area. And now about a third of my lab is working in this area. So up from one person to about a third of these people you see here. So it's really been, it was great for us. Kind of showing us, yes, we can work on this. And hopefully moving forward can make more breakthroughs and have meaning for this disorder. And I'll finish there and take any questions.
Hey, I do have a question. When you were talking about you were overexpressing the glypican-5,
Yeah.
How are you doing that?
Yeah, so the way we do this is we use a viral vector. So we know the sequence of RNA that would normally make glypican-5. So we put this in kind of a viral vector that is in a way where just an astrocyte will make this protein. And we're able to get it brain-wide because there's a lab at Caltech, Viviana Gradinaru, who identified these types of adeno-associated AAVs, this type of virus, that actually, when you put it in the bloodstream, it has special coats on the surface of the virus that allow it to enter the brain. So then when it enters the brain, it will then enter most of the cells, but because of the way we're telling the virus to make the glypican-5, it will only make it in the astrocytes. So this is a way that we can actually get brain-wide delivery. So this is something that's kind of interest to people also in biotech or for therapeutic eventually. How can you get things to the right place in order to produce the cargo that you want to make?
Oh, wow. Thank you.
Yeah.
Dr. Allen, based on your findings as a result of the CART grant, what follow-on funding did you get to move forward with what you've got?
Yeah, so we've had lots of funding, which has been just fantastic. We had funding from the Chan Zuckerberg Neurodegeneration Initiative. If you're familiar with them, they have a very big network now of people interested in neurodegeneration. So we're part of that group as well. We also are part of a group here at Salk, funded jointly by the American Heart Association and the Allen Brain Institute, which they came together because they were interested kind of in vascular or metabolic changes that could be linked to neurodegeneration. So that's how we got started working on the human astrocytes. So that allowed us to get into that area. And then recently we got funding at Salk from a foundation called the Nomis Foundation, and they're supporting our work related to neuroimmunology. And some of the things I told you about today with the astrocytes changing in Alzheimer's, a lot of it's related to inflammation and how astrocytes interact with immune cells. So this has allowed us to get into this area to be looking more at kind of some of these gain of function things that you see and how could we dampen those down. So yeah, it's been great. It was like really got us going.
Just to follow up, we have researchers struggling to figure out how to get drugs into the brain.
Yep.
And your virus delivery system is brilliant, absolutely brilliant. I had not heard that before. That gets it through the blood-brain barrier.
Yep.
And once it gets in there, then it does generate the expression of what you're looking for to deal with the astrocytes. That is amazing. Thank you for that.
We didn't develop it. Yeah. Yeah. So if you remind me or send me an email, I can send you the lab that developed that. Yeah. So
Dr. Allen, do you see much difference in when you're looking at the different times of dementia, front temporal and, MS? My sister passed away from MS. Is there a big difference between the Alzheimer's and the other two?
yeah, so we haven't looked at the timing in other disorders. What we know is just from these patient samples that if we look at the glypican 5, as those patients have more pathology, it's more decreased. And it's not just glypican 5, there's a whole kind of core set of genes, some that go down and some that go up in all these different conditions. So for us, if we're thinking of what would get a company excited about investing in therapeutics, then if it's something that you could then say there's many different conditions that you could see some benefit with this, we think those are the ones to go after first, because there's going to be more potential for more patients to be impacted.
Anyone else with questions? This has been great. Rod, yes.
Dr. Allen, in one of your very last slides, you had the term sporadic Alzheimer's. Could you help me with that?
Yeah, of course. So some of the studies you may have seen where people have been using patient samples from Alzheimer's, or even the mouse models I told you about, those are based on familial inherited forms of Alzheimer's disease. Excuse me. So the kind of, say, 5% of patients that have a mutation in the amyloid precursor protein or the presenilin gene, that they will develop Alzheimer's disease, and usually at a younger age. Whereas the majority of patients, 95%, would be considered what you call sporadic or later onset, so developing with later age, with age being a large risk factor. So like with everything, like we think the mouse models can be helpful, but they're not modeling most of the patients because most people don't have this like genetic increase in APP. So what we're doing with the sporadic is it's patients who have a diagnosis of Alzheimer's, but not having a known APP or presenilin change, and asking, well, could there still be a change that happens inside the cell linked to their risk for developing Alzheimer's that we can study in the astrocytes? So if you're familiar with kind of the ApoE4 being an increased risk factor for Alzheimer's, that's the most famous one. There are many other of these genes that researchers have identified that also, if you have a change in that gene, it also increases risk, but it doesn't mean you're going to get it. It's just an increased risk. So we want to know, can we actually model that in astrocytes to see, does that increased risk actually change the function of the astrocyte? And what we see, at least in our small samples, is that in some patients, yes, it does. So we can make an astrocyte from these patients and see it's different than someone without a diagnosis, for example.
Thank you very much. I appreciate that.
All right. Any other questions? Thank you so much, Dr. Allen. We know you are in a different time zone than we are, and you have made an additional effort, excuse me, to join us today. And we are so grateful, and we appreciate your continued support of CART. And it's always nice to hear from our researchers a few years later and see
Yeah.
what else has developed since your initial project. So thank you
Yeah,
so
thank
much
you.
for
As
that.
I say, also very grateful because it was our first grant. So it's kind of like telling us we could work on Alzheimer's. So, yeah.
We love that. Thank you.